Hello, my name is Bella Christie. I am presenting a paper that's called Understanding Pneumococcal Serotype 1 Biology Through Population Genomic Analysis. This paper was written by numerous authors, but the author that I chose to talk to was Cheryl Ann Dam. She is a professor of biology at the University of New Hampshire. The paper was published in 2016. Now on to some key words. First is pneumococcal serotype 1. This is a serotype of pneumococcal disease, which is an infection that's caused by a bacteria. And then is ST217. This is another name for the clone of the pneumococcal serotype 1 that will be used. Sequence clusters. Sequence clusters is simply the grouping of biological sequences that are somehow related, and they will also be referred to as just SC. And lastly is isolates. This is referring to the clone's DNA that had to be isolated. Now is the hypothesis. The basic hypothesis is that the clone evolved in different ways in different places, and that was really the only hypothesis that they had, because the study was more to just understand the biology behind this clone, but they went into it looking for this specifically as well. Now on to methods. First, they had to isolate the collection. So they collected these isolates over a range of years, and once they were all collected and genomic DNA libraries were formed from the isolates, they were all sequenced. Next, they had to detect the serotypes and sequence types, and to do that, they used PCR. Third, they had to detect recombination and construction of the phylogeny, and they used GATK to realign the insertion and deletion sites, then generated consensus whole genome sequences. From there, sequence clusters were identified. Then, within sequence cluster genetic diversity, to get that, they calculated the pairwise distances between isolated in the phylogeny and the terminal branch distances. Then, to find the most recent common ancestors and the mutation rates, they investigated the temporal evolution patterns within SC. Then they had to detect presence of antibiotic resistance genes, and those were obtained from GenBank. Finally, they did some statistical analysis, and all those tests were performed using R v3.1.2 and GraphPad Prism v6.0. Now on to results. This figure shows 5 distinct cascades, and in part C is the association between the continent of origin and the ST217 isolates with sequence clusters. And you can see that is the red and the blue. And the blue is the Africa continent, and the red is for Asia. In part D is looking at sequence clusters, and it's looking more at by country rather than by continent. Here in the second figure, it's very similar to the first, but actually very different. They identified 1,520 core genes present in at least 99% of the isolates, and an overall gene repertoire of 4,594 genes. They also showed that African isolates showed higher prevalence in a certain test than the Asian isolates. This figure number 2 shows it by sequence cluster, continent, country, and then in section 4 of it, it shows if it has antibiotic resistant genes, and if it's red, the genes are present, and if it's blue, the genes are absent. So for most of them, most of those antibiotic resistant genes are absent, and you can see it's ordered by sequence. So for sequence section 3 is the green. If you look over for TETM, most of those are present in that sequence cluster. And it's just another test that they ran to test the different sequence clusters and how they formed antibiotic resistance to certain genes in one area as opposed to genes in another area, which is interesting just to show how the gene evolved. Then in figure 3, this is basically comparing the resistant genes again and elements in different sequence clusters and countries. So first you can see in A you can see the resistance genes in Africa and Asia, and you can see 2 different resistance genes in Africa and Asia. And since Africa is the blue, it has a higher proportion than Asia. Then in B, C, and D, those are all by sequence cluster. In B and C, it's looking at sequence clusters and other antibiotic resistance genes or elements. And then in D, it's looking at a deletion of a certain antibiotic resistance gene to see if those are similar or if they're different based on the region or sequence cluster. Before getting into what they actually found in this study, I wanted to talk about why this study is so important. This data set that they have offers a unique opportunity for the understanding of how the clone has evolved and spread. And this is a highly invasive bacteria that can cause horrible things, so it's important to know its makeup and biology, and basically anything you can know about it, you would want to know about it. This disease kills over 1 million children annually. And that's why it's so important for us to know as much about it. Now, on to what they found. Their findings showed evolution of the clone into geographically distinct lineages with different characteristics. So, the evolution of the clone in different geographic areas had different characteristics. The findings show with higher genomic resolution of the recent spread of the clone. The findings also show intracontinental as well as intercontinental spread of the clone with high resolution using whole genome sequencing. Their findings showed consistently lower than expected amount of polymorphism under the mutation drift, which is interesting because they had expected to find a lot more than what was actually there. Since the ST217 clone has a high phylogeographic diversity, which means it has a wide spread of places that it has been, and it has a wide spread of characteristics. The clone has been in circulation for a long time globally, and they knew that because of its high phylogeographic diversity. This allowed diversification and adaptation in many geographic regions. When I talked to my author, she said that they actually needed to do further studies to completely understand the biological mechanisms that make the ST217 clone as highly invasive as it is. She also mentioned that there was a potential limitation that occurred. They didn't gather the same number of isolates from all countries. It was varying. So that could have affected it. However, they tried to avoid that being a limitation by focusing on sequence clusters rather than by individual countries. And while on some things they did show the individual countries, they also showed the sequence clusters, so they wouldn't they didn't let the countries go without the sequence clusters, just in case that proved to hold back the information that was actually there. And she also said that nonetheless of a possible limitation or any other mistakes that could have occurred, or the fact that they have to do more studies, that this study provided a comprehensive genomic portrait of evolutionary dynamics of the clone. Thank you.